This is a video about modeling based on a PowerPoint presentation by Damien, Batstone. First, what is a model? Well, it's a representation of a system in some other form than the system itself. There are three main groups of models: physical, conceptual, and mathematical. We focus on mathematical, but we'll also discuss the others. We do modeling because it's very useful. It's useful for the operation of treatment plants of various kinds, get expert opinions, evaluate, get familiar, just training, and test conditions that are beyond what you can do in the real case. For research, it may be even more useful. Use it to really find out what's going on, maybe design experiments, understand experiments, and also to share information when discussing, presenting, and publishing. This we may call a conceptual model. It's the basis model, modeling based on mass balance. So first we define a system boundary, we see what goes in, what comes out, and what's going on inside. And part of what's going on inside is reactions. So here we have different terms we can use, but there are two main considerations: the stoichiometry, in other words, how much do you get of a product from something reacting, such as biomass from feed. But also how fast it goes is necessary to know to make a good model. If we look a little bit closer at how we do stoichiometry, there are different ways of doing that, but certainly more and more we use chemical oxygen demand as a basis, because that's more or less a direct measure of energy or electron transfer. But the reactions also must be balanced in terms of the elements and charges. Now then, since we have models with different types of components, we use yield as a way to compare or express how different components in the model interact. And since we mainly work on biological processes here, the reaction terms, they often combine anabolism and catabolism, meaning anabolism is where you take the feed and make some product, typically to recover energy from the feed, and then anabolism is building new biomass based on the same feed. And overall, therefore, the substrate goes to products and biomass. We can model this in two main ways. We call it either uptake models or growth models. The uptake models, where we base it on anabolism, saying that the cell growth is related to the rate of substrate uptake. Growth rate mu is equal to the yield times substrate uptake rate. That's one way of doing it. The other way is growth model, where rate of growth is related by a yield. Essentially the same thing, but it makes a difference for how easy it is to deal with the equations, especially when we have many equations in a complicated model. Here we see generalizing uptake and growth on the chemical oxygen demand basis. One COD substrate goes to one COD product, or a fraction of the substrate goes to a fraction of biomass. And then we have the growth-based, where we say use the yield terms for the catabolism, while the anabolism then, the yield is one substrate goes to one biomass. Uptake based, as we can see, a little bit more complicated due to the way the yields end up being in both catabolism and anabolism. Okay, we go on and we look at the typical case where we have a continuous flow stir tank reactor. We know what goes in, a flow and the concentration, and within the volume V, we have a concentration that is equal to the concentration coming out. And since it's a CSTR, we assume it's homogeneous. So in other words, whatever is inside here is also represented by what comes out. And the equation, mass accumulated with time, is based on what comes in, what goes out, and the mass produced or consumed. Typically, we try to simplify. In most cases, volume is constant, so we can put that outside of the integration, and we end up with relatively simple equations where a change in concentration with time is related to dilution rate or flow divided by volume, concentration in and out, and again the reaction. And at steady state, it's further simplified since the left side becomes zero. Now, if we look at kinetics, we typically assume that, okay, so difficult to read, but it's when the concentration of substrate is low, the rate is a linear function of concentration. But as substrate concentration increase, it kind of levels off, and in the end, if we add more substrate, there will be no higher reaction rate. And this is called either Michaelis-Menten, after the people who first discovered this in enzyme reactions, but then it's often called the Monod, since Monod was the person who saw that this is also valid for bioreactions. So here we have it more clearly expressed. Michaelis-Menten for enzyme kinetics, and Monod is when we have biomass and bioreaction, and we see the terms are essentially equal, but when we have a bioreaction, we multiply by the number of microorganisms. This is also called the saturation function. If we go on to other reaction terms, we have death or decay. There are always some organisms disintegrating. There's also hydrolysis of particles, and these are both all typically modeled as first order. So just saying that the reaction is proportional to the amount of reactant in the process. There are also many things that can inhibit a biological reaction. So then we have an inhibition term, which we usually then just multiply by the reaction. So inhibition, if there is this term, if there is no inhibition, it has a value 1, so it doesn't change the reaction. The stronger the inhibition, the closer it gets to 0. And it can be influenced by temperature, pH, nutrients, products, even substrate can inhibit in some cases, and certainly toxins. All this can be put into a stoichiometric matrix. So if we look at the equation we had with CSTR, it can also be expressed in a matrix like this, the components, in this case substrate and product, and some rate. And first we just assume there is one reaction, substrate is consumed and product is generated at some, in this case, linear rate. So it can be expressed in this way. And if we put this together, we end up with two mass balances, one for the substrate and one for the product. And there are several standardized models in environmental engineering based on these principles. It's taken a long time to develop these, but it's a really useful way that there's become standard models with standard parameters, making it much easier to work on and communicate between researchers and practitioners. Most of these models are developed by the International Water Association and published in similar reports. So let's take a look at a little bit of this. First of all, where most of the work has been going in is in activated sludge, since that's the most applied biological process for wastewater treatment. Started out with activated sludge model number 1, mainly looking at carbon oxidation, nitrification, and denitrification. Then this was extended to ASM2 by also including phosphate storage or phosphorus removal, biological phosphorus removal. And then this has been, as new discoveries were made regarding these reactions, modified versions of these two has been introduced, and this ASM2D is probably the most popular at the moment. And then there is also an ASM3 that has taken in a somewhat different perspective on how the cells are decaying. So rather than saying decay, we're saying they're requiring substrate for respiration. Other standard models, ADM1, anaerobic digestion model number 1, very important, very useful. And there are a few other models, but especially ADM1 has become a very important model in environmental engineering. The ASM1 came already in 1987. It's based on biomass growth, as we discussed earlier. We use monokinetics or saturation kinetics for growth. It has two different main components, kind of, using COD and nitrogen in as different states in the model. It's, in this context, relatively simple with 13 components and 8 processes. And then if we put it into a matrix, it's a 3x8 matrix. And here we can see how it is implemented in a typical activated sludge plant, where we have the inlet flow, we have the reactor with 2 compartments, one anoxic for denitrification, one aerobic where air is supplied to stimulate nitrification. And then the flow out is separated by sedimentation and biomass is returned. There is also returned flow from what we can say is the aerobic to the anoxic zone to bring the nitrate generated here back. So if we start here, we see the aerobic growth, the heterotrophs. It takes the COD with oxygen and makes biomass. But it also has this autotrophic reaction, taking ammonia with oxygen, making nitrate by another type of organisms called autotrophs. And this nitrate is what then needs to be brought back to the anoxic zone, and that's done through this line. So in here, this same nitrate generated downstream can react with COD that comes on the inlet, and we get what we call heterotrophic denitrification. So we turn the nitrogen in the wastewater into nitrogen gas that is stripped off into the atmosphere. And then there are also some hydrolysis where COD, larger molecules, are broken down, and we have the decay of both the autotrophic and heterotrophic biomass. This can, if we put it into a matrix, it looks like this. Okay, now this becomes a little bit too much for the screen, but you can study this on your own at some point. But we have the different 13 components and the 8 reactions we mentioned, and here is the stoichiometry, and here we have kinetics, the reaction rates. Okay, then we go to the next main model, which is the anaerobic digestion model number 1, where we have organics such as activated sludge that is taken off from a wastewater treatment plant, going through various reactions and in the end producing methane, and thereby getting rid of excess sludge and turning it into a useful product. We see there are some steps here where we have particles being broken down and hydrolyzed to sugars, amino acids, and long-chain fatty acids. These are small enough molecules that they can be taken up by microorganisms. And we see here that we have 7 different microorganisms doing different reactions. So sugars are turned into valerate and butyrate and acetate and propionate. Amino acids go down here to acetate and hydrogen, and long-chain fatty acids also go to hydrogen and acetate. Acetate is further then broken down. Okay, these 1, 2, 5 are bacteria, and then acetate is taken up by methanogens, which are different types of organisms called archaea. 6 here, they turn acetate into methane. There's another group of archaea that can take hydrogen and combine it with CO2 and make methane. And then we see that all these different organisms can also die, and if they die, they become food again, complex particulates. Okay. This model, even if it's the newest, it's widely used. It's based on substrate uptake. It uses monokinetics for growth rates. It treats organics together with organic nitrogen in a good way. It has 26 components and 22 processes, so therefore the matrix now becomes more complicated. Also, it contains physiochemical processes such as acid-base reaction, gas stripping, making it very useful at looking at, for instance, what happens to nitrogen and what kind of inhibition can you get, when will it work. So ADM1 is a key model for environmental engineering these days. Whether it's wastewater treatment or making biogas from food or manure, there are so many applications of this model. So then we can say that there are these standardized models. They were initially just made mainly for research, but it's really been, when they became standardized, they have become useful in many different ways. But there are also some standardized packages that can be included and used. There are also specialized models for hydraulics, like how the flow is when you don't have a CSDR but more complex systems. So these are the reasons and how we use these standardized models. But some of these commercial models are also more graphical and more easy to use. So for instance, here we have an example, BioWin, or EnviroSim it's called, where you put in all the different steps of a big wastewater treatment plant, and you can simulate everything, and you can put some of these standard models we've already discussed into the different boxes. Like here, modified activated sludge model type that can be put into the various boxes. There are more examples. West, same difference really, but where you can use the different activated sludge models and so on, but also other types of models we mentioned before. So once you have that, you can also do different packages, like you can do simulations, you can follow what happens over the day and night with high loads, low loads. You can look at nitrate, ammonia, different stages. You can simulate by turning the aeration up and down or on and off, just like you could in a full-scale plant, and then look at the effects. So you can really simulate situations where that you cannot really test without putting too much risk into a full-scale plant. You can test the conditions using your model. There are some other considerations when you work on this. One of them is how to calibrate. Some parameters are local, but in general, and most of the time, some of these parameters like growth mu max and all these what we can call fundamental or intrinsic coefficients for the different organisms, they never change. So that therefore never touch them, such as those that go into the Monod rates, growth rates. On the other hand, hydraulics, of course, can change a lot. And but most of the time we work in mixed tanks. Sometimes we do plug flow, sometimes we do aggregates, such as biofilms. But as we move to the right here, the model becomes much more complicated in terms of numerics and everything, but also very useful. So, but what we can change and what does change in real life is the influent, whatever is coming to the plant. It changes over the day, week, year, and that is also something that you can change in the model to see what happens. Will it handle the winter conditions, for instance? And then the final message, modeling is getting easier due to the fact that they become standardized. We've shown a few examples here. And the packages available are quite easy to implement and use. But still, you should really know what you're doing. Otherwise, you can get all kinds of nonsense. So therefore, you really need to study well to know the fundamentals of what these reactions are and what's going on. And if you have this process knowledge, then you can also use the model in a very efficient way.